Thank you very much for the introduction. I'm going to talk about some of the regulatory considerations to establish API sameness in complex APIs. This is the outline what I am going to talk next 20 to 25 minutes. I will provide a brief introduction about the requirements of API sameness in generic drug products and the challenges to determine API sameness in complex APIs. The recommended approach to establish API sameness in complex APIs with 3 examples. We know that by regulation, a generic drug product must be therapeutically equivalent to a reference listed drug. As shown here, theoretical equivalency of generic drug product is dependent on pharmaceutical equivalence and bioequivalence. Further, the pharmaceutical equivalency depends upon if it has same API, same dosage form, and route of administration, identical strength, and concentration. So, API sameness is required to obtain pharmaceutical equivalency and theoretical equivalency of generic drug product. So, what is same API? It is considered same API as RLD if it possesses same physical and chemical characteristics as RLD, same stereochemical characteristics, same polymorph where relevant, impurity profile for current regulations, and compared with RLD where required. And how the API sameness is determined? API can be divided into 2 groups like simple APIs and complex APIs. Simple APIs are small molecules with a defined molecular weight. These molecules can be characterized by conventional methods such as NMR, MOS, IR, etc. Impurity profile also can be determined by traditional analytical methods such as HPLC, GC, etc. So the example I given is simple molecule here, diazepam. In other hands, the complex APIs are heterogeneous mixtures with no defined molecular weight. Complex APIs cannot be fully characterized with traditional methods and requires product-specific analytical techniques. Due to their properties, establishing impurity profile also challenging. Complex drug substances have become more common in approved generic pharmaceutical products in the past several years. So what is the approach for these complex APIs to show API sameness where traditional methods won't work? Due to the properties, it may not be possible to characterize entire molecule with any single analytical method. Each structural fragment is analyzed with different techniques and compared with the structural fragment of RLD. So orthogonal characterizations are required to fully characterize the complex API. In addition to the orthogonal characterization, the enhanced manufacturing process development is also used to identify critical quality attributes of the API and establish functional relationships of raw material attributes and process parameters to critical quality attributes of API. Since the identification of impurities in the final API is challenging, appropriate controls for impurities may be needed in the starting materials, intermediates, and other raw materials used in the process to avoid formation of side products. Additionally, in vitro biological and functional assays are used to establish the API sameness. The justification of analytical methods used for characterization is needed where required. To establish the API sameness of complex APIs, the totality of evidence approach is used. In this approach, the sufficient structural signature analysis, manufacturing process information, and functional data are compared to demonstrate the quality of the API is highly similar to the API in RLD. In this slide, I show some examples of complex APIs. Complex APIs include synthetic polymers with heterogeneous mixtures of small molecules such as Colesevelam Hydrochloride, naturally isolated compounds such as conjugated estrogens, semi-synthetic mixtures such as Enoxaparin, peptides such as Linaclotide, and also macromolecular complexes and synthetic nucleotides and others. In this presentation, I'm going to talk about the strategies used for the API sameness for these three examples. Due to time constraint, I cannot talk in detail, but I will give you high-level overview how API sameness can be determined using the totality of evidence approach in each case. Here is the first example, Colesevelam hydrochloride. Colesevelam hydrochloride is bile acid sequestrant and used to treat hyperlipidemia. As shown in the structure here, it is a cross-linked polyalylamine and alkylated with two types of alkylating agents. The API is insoluble in a solvent, and it is a heterogeneous polymeric mixture. The drug is not absorbed. The intended scavenging of bile acids happens in gastrointestinal tract and excreted from gastrointestinal tract unchanged. So it is challenging to characterize using traditional methods and recommended totality of evidence approach for establishing the API sameness of this API. Based on the information provided in the product label, the Colesevelam hydrochloride can be typically manufactured by cross-linking polyalylamine with epichlorohydrin, followed by alkylation with two alkylating agents. As seen in the final structure, it has four types of structural fragments with A, B, C, and D. The process looks simple, linear manufacturing process, but the final API is little complex. Then how to characterize this final API to show API same as RLD? The structure signature analysis and process parameters can be used to establish these four fragments are similar to the RLD. The basic backbone of the structure comes from polyalylamine, and the properties of the polyalylamine may affect the critical quality attributes of the final API. Hence, the quality attributes of polyalylamine, such as molecular weight, impurity profile, should be appropriately controlled in polyalylamine. The structural fragment B is a degree of cross-linkage. Since the final API has several components, it is easier to establish degree of cross-linking at the intermediate stage. This is expected to carry forward to final API. The amount of epichlorohydrin used to obtain the structural fragment B in proposed reaction conditions should be established. The degree of cross-linkage can be estimated using deconvoluted solid-state carbon-13 NMR. The further characterization of this cross-linked polyalanine can be done using elemental analysis, total titratable amines, DSC, TGA, swell index, etc. All these tests further support the degree of cross-linkage. To obtain appropriate structural fragments C and D, the process conditions of the reaction should be established, such as reaction time, amount of each alkylating reagent, amount of base used, timing of the addition of these raw materials, etc. Impurity profile of the raw materials and intermediates can influence the impurity profile of API. Due to insoluble nature, the API, the impurities formed also may be insoluble and cannot be identified if they are formed. Hence, proper controls are required for all the intermediates and raw materials to avoid getting unintended side products. With process development, the amount of raw materials are required for fragment B, C, and D are studied. The final API further compared with RLD with structure signature analysis using orthogonal characterization of final API and API extracted from RLD. The characterization methods used are carbon-13 NMR, FTIR, Raman, elemental analysis, total titratable amines, swell index, etc. Equivalence of physical properties should be established, such as thermogrammetric analysis and difference scanning calorimetry, particle size distribution. Related substances such as soluble oligomers and PJ should be evaluated. The in vitro bile acid binding capacity can be used to show the API sameness. Here the 4 bile acids should be used with, and the total binding capacity also can be used. Compare multiple batches of test and referenced API. As discussed, to establish the API sameness of Colesevelam hydrochloride, totality of evidence approach is used with a robust manufacturing process, orthogonal characterization methods, and in vitro functional assay. There are 2 published product-specific draft guidances for Colesevelam hydrochloride, one for the suspension, another one for the tablets. Now I jump right into the next example. Enoxaparin. Enoxaparin is a low molecular weight heparin which has antithrombotic properties. As shown here, it is a heterogeneous mixture. Unlike previous examples, this is very soluble in water. Low molecular weight heparins are structurally complex, highly sulfated and negatively charged linear carbohydrate polymers. The average molecular weight of this enoxaparin is between 2000 and 8000. The characteristic of enoxaparin are the double bond in disaccharide unit, and the reducing end has around 20% of 1,6 anhydride derivative. Low molecular weight heparins are prepared by chemical or enzymatic depolymerization of heparin, as shown here. Identify critical quality attributes of API. Establish ranges for critical process parameter to achieve the comparable critical quality attributes of API. Heparin can be extracted from different sources such as bovine and porcine. The clinical activity may be similar for the heparin from different sources, but there are few differences in the orientation of disaccharide building blocks due to difference in biosynthetic pathways. For the consistent chemical composition and mapping, the source of starting material should be established based on the comparative studies with RLD. Depolymerization also can have effect on the disaccharide units, especially on the end units. I shown two different depolymerization methods give two different low molecular weight heparins. Though the clinical activity of these two low molecular weight heparins is similar, their chemical composition is different, especially the terminal building blocks. The depolymerization method with proper process controls should be established for the consistent quality to obtain the API which has comparative properties as RLD. Negative controls also can be used in development studies to establish controls and ranges. After establishing the required starting material and manufacturing process, how to determine the end product is same as RLD. Enoxaparin characterized by structural fingerprint analysis and physicochemical properties and compared with the API in RLD to show API sameness. The structure signature analysis study involves enzymatic cleavage, chromatographic separation, mass spectrometry, 1D and 2D NMR, etc. With these studies, equivalency, oligosaccharide composition, sequence of oligosaccharide species, fragment mapping, etc. should be established. The structure signature analysis also useful to establish the source of starting material. Equivalency in physical properties such as molecular weight also should be established. The in vitro coagulation properties of the API can be used to further support the API sameness of enoxaparin. You also need to evaluate the impurity profile to make sure there is no increased risk in your generic copy of API. As discussed, to establish the API sameness of enoxaparin, totality of evidence approach is used with the robust manufacturing process, orthogonal characterization methods, and in vitro functional assay. There is published product-specific guidance for enoxaparin. You also can refer to Nature Biotechnology publication listed here for details. Now I jump right into next example. The third example is linaclotide. Linaclotide is agonist of guanylate cyclase 2C approved for the treatment in adults of constipation, predominant irritable bowel syndrome, and chronic idiopathic constipation. As shown here, linaclotide is 14-mer with 3 disulfide bonds. Unlike previous two examples, this has defined molecular weight and solubility in water. Still, it is a complex API based on the characterization methods needed to show API sameness. The sequence of the peptide stereochemical properties depends upon the manufacturing process and selection of the starting materials. The sameness of the active ingredient of proposed generic synthetic peptide can be established through physicochemical characterization and biological evaluation. The final API should be further evaluated by appropriate analytical methods to show the peptide has intended sequence and stereochemical properties. In linaclotide, there are 3 disulfide bonds, and the configuration should be established with appropriate analytical techniques. The impurity profile also important. The equivalency can be provided with comparing RLD. The impurity profile is also should meet current regulatory requirement, such as meeting the requirements in general guidance for the peptides and product-specific guidance for linaclotide. The equivalency in biological activity of the API can be used for further establishing the API sameness of linaclotide. As discussed, to establish the API sameness of linaclotide, totality of evidence approach is used with robust manufacturing process to get 3 disulfide bonds with appropriate stereochemical properties. Orthogonal characterization methods to characterize the peptide and in vitro biological for binding and functional assays. In conclusion, totality of evidence approach is stepwise process to determine the API sameness for complex APIs. In these 3 examples, the techniques used are different, but use totality of evidence approach. No single component can establish the API sameness in case of complex APIs. Each complex API has its own challenges as discussed in previous slides. DMF applicants should evaluate individual situation and apply the recommendations and required principles accordingly. Identify critical quality attributes and establish ranges for critical process parameters in manufacturing process through developmental studies. Develop negative controls and use them to establish ranges for critical process parameters. Characterize multiple lots of the active ingredient mixture for both proposed generic drug product and reference product under similar conditions. At least 3 batches are recommended. Provide all the required data and literature support for the analytical methods used for the API sameness. Look for the product-specific guidance. In the next slide, the information on where to look for guidance is provided. For some complex APIs, there are some compendial monographs. Follow the recommendations in the guidance and compendial monographs. There are options available for applicants to request pre-development meeting and pre-submission meetings. There is also an option for teleconference to clarify the deficiencies if required. This page provides the resources for looking for guidances and information for requesting meetings. Check also DMF website for the information. I work with mainly Ram and Ben on complex APIs and also with David, our division director. Mani and Kedu helped me to build the content for this presentation. Thank you. You may also get additional information in these listed poster presentations here. If you have any questions, please type them in Q&A box below. If you have any questions not answered during this workshop, you can send them to given email address on the slide by March 15th.